Hi everyone, my name is Tracy Hart. I'm the CEO of the Osteogenesis Imperfecta Foundation, but I am the co-chair of the Coalition of Patient Advocacy Groups Steering Committee. My other co-chair, Kirsten Wheeden, is one of our presenters today. And we wanted to welcome you today to the second session in our webinar series, which is clinical trial readiness from the patient perspective is the overall title of the webinar series. And today we're going to be talking, our title is Working with Your Patient Community, and we're specifically going to be talking about data. And data as it relates to, how important it is to have really good data, what our data means as patient advocacy groups and how valuable it is. And then we're going to hear from two of our colleagues that will share how having that really great data transfers to having good outcomes with clinical trials, and maybe some challenges as well that they'll talk about. So I'm going to introduce the presenters in order of appearance. So the first presenter will be Tiina, and most of us know Tiina, but she is the program director for the Rare Diseases Clinical Research Network at the NIH. And as the lead, she collaborates with 10 NIH institutes, 20 plus consortia, the Data Management Coordinating Center, and all of us. So she has a huge job. So she's going to be our first speaker. Our second one is Eileen King. She's professor in the Department of Pediatrics at the University of Cincinnati College of Medicine in the Division of Biostatistics and Epidemiology at Cincinnati Children's Hospital Medical Center. And she's the co-PI for the Data Management Coordinating Center. Our third presenter will be Josh Tarnoff. It says Joshua here, so Joshua Tarnoff. He's the CEO of the NephCure Kidney International, and he's representing the Neptune Consortium. And then our last speaker will be Kirsten Wheeden. She's the executive director of the American Porphyria Foundation.
Got it.
Did I get that? I always
got it. Thank you.
I've known you long, and I still do that. And she's representing the Porphyrias Consortium.
So my name's Tiina, and as Tracy introduced me, I'm sort of all things RDCRN in this point in my life. Very happy to have this job and feel very lucky to be working with you all. So first slide, please. What I'm going to talk about today is let's look at the landscape of where rare diseases are and where we are in the drug development landscape. So it's a different landscape than the drug development landscape in the general disorders. And we have a lot of people, if you look at this, working on their own islands, not necessarily speaking the same language. So if you look at the map, you'll see people are working on their registries, people are working on the science, and we all have the same goal though. We're all trying to find a treatment for rare diseases. Next slide, next click please. But what we're doing is we're working in silos. And so when you're working in a silo, all you can see is the end of the tunnel. And we're making the same journey and we're walking the same path. Next slide, please. So we're in this silo. We're all looking at one point, our own point, and it's very important to us. But we really need to think about working together. Next slide, please. Because what we're really trying to do is we're all on the same journey. We all want a treatment for our rare disease. So what's the holdup? Next slide. So one current estimate right now is it can take 10 to 15 years to get a drug to market. It can cost about $2.6 billion to develop a drug from the initial drug discovery to completion, and fewer than 12% of approved drugs entering development get approved. And the current approach we have right now, I think we can all agree, is not sustainable. Next slide, please. What do we need to do? We need to work faster. We need to work in a more cost-effective or cheaper manner. The data needs to be high quality, and we also need to have what we're doing be sustainable. Next slide. So how can we work faster? So We can establish networks, which is something we've done. We've brought the people together that need to work together. We need patient advocacy, and we've brought those people to the table. You're really embedded partners in what we're doing. We need natural history studies, and tools to need to be established. We need to establish outcome measures, biomarkers, common data elements. Those are the types of things that help us work faster. Next slide. We need to work in a more cost-effective manner because we can't afford to keep working the way we're working now. We need to work in a shared environment. We need to share tools. We need to be innovative in thinking of different models and for trials like basket trials and umbrella trials, and people can talk about that later, but those are just different statistical models or clinical trial models that can be used. Next slide, please. And we need to work to have higher quality data. And if we go back to that first map that we're looking at, that was developed when Ann Pariser and I were working on an HHS program to look at what does the landscape look like right now for rare disease drug development. And we interviewed people from researchers, we interviewed family groups, we might have interviewed some of you, we interviewed people from the FDA, people from small industry, from small pharma and big pharma, and from investors. And the one point that they targeted on was data, that one of the biggest holdups and one of the biggest problems that we have are poor data that are being developed from the very beginning of the route to the very end. So we need to have data standards to help have higher quality data. We need to use good data practices. The research we do needs to be rigorous, and it needs to be reproducible, and it needs to be transparent. So we really need to, whatever we do needs to be of high quality. Next slide. And it needs, if we're able to work faster, and if we're able to work cheaper and have higher quality products, that leads to sustainability for moving through this process that takes such a long time to develop a drug. Next slide. What is the big thing that we're trying to get rid of? We're trying to de-risk this whole process, because that's one of the biggest problems we have when you're going into drug development. There's a lot of risk, so we're trying to erase some of that risk. Next, click. And the best way to do that is to be a good scout and be prepared. And so we made a lot of changes in the RDCRN in this cycle, the Rare Disease Clinical Research Network. And a lot of those changes that we made in the activities we're doing, how the DMCC was changed around, how we're bringing the CPAGs in to be involved at a deeper level, it's to de-risk this process and be more prepared when we reach the time that we're going into a clinical trial. Next slide, please. So clinical trial readiness is really the de-risking process. It's the what are we going to do, where are we going to do it, when are we going to do it, who's going to be doing it, how are we going to do it, and why are we going to do what we're doing. So the more prepared you are when you're going to a clinical trial, the less the risk is. You're not making a risk. You don't have the risk of when is the best time to treat a condition, or do we have everything in place, or who do you treat? How will we conduct the trial? Who will conduct the trial? The whys, what answers are we trying to get when we're doing our research? The what is the best potential research we're doing? That's where the natural history information comes in, and that's why it's important that we collect really good data. Next slide. So what we've done in this new iteration of the RDCRN and the DMCC, which is RDCRN4, is really created a cloud environment. And basically, we've built a platform, which is the basic infrastructure. And those are the basic services. That's the cloud services that we're using. And we happen to be using AWS. On that platform, where everyone will come to work, is a managed platform where we have all the data security, we have data storage and data standards. And those are the kind of things that you might not be seeing every day, but they're there to protect your information, your data, and to help manage and have the strongest information and the most protected information we have. And then there's the managed services that the DMCC are providing that are tools that you can use in this cloud environment. Next slide. So we've brought all the consortia into the cloud environment. Next slide. And we've given them lots of tools. We've given them data tools, resources to work together, a workspace to work together. We're working on preparing data standards, and Eileen is going to talk about that. So what you're doing is not reinventing the wheel with everything, because basically everyone has to follow that same path from beginning to end to get a drug developed. And working together, we have more leverage, and we're able to do it in a more cost-effective and faster manner. Next slide, please. So some of the questions that have come up with, you know, why are you making these changes? We've been doing this way for 15 years. Why change it? Those other rare diseases have nothing in common with my group other than being rare. Why are we doing this? How can we possibly work together? Next slide. You know, people will say, this is a natural history study. It's not a clinical trial going to the FDA. Why do we need to bother with good data standards and good data? And, you know, some of the investigators might even say, I don't want to share my data right now. You know, what if somebody scoops my science? So next slide. So what we really need to do is find commonalities across the rare diseases. And that's one of the things we talk about a lot. You know, we all want to have a successful clinical trial now. Everybody can agree to that. We all want to collect data and analyze it and have it stored, and we want it to be safe. Everybody can agree on that. We all want to remove as much of the risk of failure as possible before we go into a clinical trial. These are things we all have in common. Even though we're looking at different rare diseases, we're all working towards the same goal for our own individual purposes, but we're all doing the same thing. And we want to our work to be meaningful across time and to be saved. Click. So what we need to do is we need to take off our individual rare disease hat, or our individual consortia, or our individual specific disease hat, and put on our rare diseases hat, where we can all work together and agree on similar problems, and we'll have a stronger voice to do that. Next slide. So let's put on our zebra thinking caps and think about what can we do to work faster, to be cheaper, to have high quality. So what we're doing is sustainable. Next slide, please. So I guess my take home message today, and my very quick overview here, is always keep the big picture in mind. It's when you're working in the field of rare diseases, it's not just your own individual rare disease, it's multiple rare diseases, and by working together, we can work together faster. So think of ways that we can be more nimble in what we're doing. Think of ways that we can make things more economical and demand high quality research and data at all stages. It doesn't matter when you start collecting your data. This data could in effect end up going to the FDA. It could go to an investor. It could go to industry. And what they want is strong data, so they don't have to repeat it. They don't have to replicate it. They can just keep moving on instead of taking a step backwards to replicate the data to make sure it's good and whatever you've done is sound. And collaborate. We can't do this alone, and working together is very important. Next slide. So I like this analogy of a snowflake. Alone, each rare disease is individual, it's unique, and we all feel that way every day, that we're special, we're unique. But a snowflake by itself is kind of weak and can melt. But if you think about it, if you bring a lot of snowflakes together like a glacier, you can basically move mountains if you work together. So next slide. So I just want to thank the program directors, scientists that we work with, the patient advocacy groups who inspire me every day, the DMCC who enthusiastically embrace our vision for faster, cheaper, higher quality that we're, trying to get at. And that's basically what clinical trial readiness is. It's be ready, be able to work quickly, be able to do it in a cost-effective manner. And the best way to do that is share resources and work together where you can. You don't have to lose your individuality to do that, but working together is really important to develop high quality information that can be shared. And I thank Mira, even though her computer froze on me.
Yeah.
And my colleagues at ORDR. So thank you. Sorry, it went a little bit long.
Yeah, you're great. Thanks, Tiina. That was a really great overview. All right, I'm going to turn it over now to Eileen King. And Eileen, you're going to talk more with us about data.
So thank you. I'm very excited to talk to you today. I want to talk about the importance of high quality data and standardization for valid decision making. And there's going to be a little bit of a statistical twist on this. So just wanted to let you know that. Next slide. I like to show this slide every time I give a presentation to really keep us focused on what we're trying to do. We're trying to improve the health and wellness of patients with rare diseases and their families. Hit again. And what we really are focusing on is we want to have faster diagnoses, so patients are not in that diagnostic odyssey, and we want better treatments so people can improve their quality of life and live more normal lives as much as possible. And our two strategies this time in this particular cycle, as Tiina talked about, was data sharing and clinical trial readiness. Hit again. And both of these, hit one more time, both of these are very important to hit our goals of having faster diagnosis and better treatment. Next slide. What I really want to get across is patient participation and commitment is the essential component for clinical research studies. So this is just an example. So we have patients or participants who are willing to engage in our clinical research studies. And of course, the participants are the key component of it. We can't do any clinical research without our participants and our patients agreeing to participate. And the participants that we have in our clinical research study really dictate what kind of decisions we can make about what kind of patient populations. So it's really important that we have a great representation of the rare disease community, of the particular rare disease patient population when we're trying to do a clinical research study. Because this really dictates who can we make inferences about, who can we make decisions about. So we really strive as much as possible to have a good representation with regard to sex, with regard to race, with regard to many other different characteristics of patients, so that we, like I said, can make valid decisions. And it's important that these particular individuals stay committed to the research study, because when they aren't committed or they decide they no longer want to participate, this really does affect the quality of data that we have. And when you are affecting your data quality, you're also affecting the kind of decisions that you can make. Let's go ahead and hit this a couple times, and or at least one more time, and maybe one more time. So again, high quality data and complete data are essential for valid decision making. So I wanted to get across what happens when participants do not agree or do not decide to continue in a clinical research study. So for example, and I don't know if you, so for example, you'll see that I have some black X's here. And what this means is that either these participants didn't come in for these particular visits, or it means these participants did not come in for these particular visits. So when we're trying to synthesize the data for these participants, we don't know what would have happened to these individuals on whatever we're measuring at these particular time points. And let me tell you what has to happen in the statistical sense, because your statistics is really helping to drive your decision making. We have to impute these data for these particular patients, because in a clinical trial, we typically talk about having an intent to treat population. And that means any patient who begins the study, we have to deal with them and their data and their missing data throughout the entire study. So for example, if a patient quits a study early or doesn't contribute data at a particular time point, there are various approaches we can use to impute their data. One is to look at patients that have similar characteristics as that patient that didn't contribute data. And we look at what their data were at these particular time points, and we impute and we say, well, we're going to have to assume this participant would have provided data very similar to patients like them who did continue to participate. That could be a very strong assumption, but that's kind of the best we could do. If your outcome is actually a dichotomous outcome, and so, for example, you're trying to see, does your drug work to have a successful outcome or a failed outcome, like does it keep a patient in remission or not? And if at your primary time point we're missing data for that particular person, the regulatory agencies will often say, well, if your patient didn't complete the study, we are going to consider that they are a failure for that particular endpoint. So what I'm really trying to get across is high quality and complete data are really essential for valid decision making. And it's really key to try to keep your patients engaged and for the patients to really recognize how important it is that they're engaged. And also for these unique patients to really participate in clinical trial research, because if we don't have a good representation of a minority population, we're not making good decisions about how well a drug or an intervention would work. Next slide. What are some of the sources that we get for our data for clinical research studies, and how might this impact our ability to make decisions? Excuse me. So many times in a clinical research study or a natural history study, participants actually go in, they have in-person visits with clinical research staff. In this case, it's much easier to be able to standardize how the data are collected, what the processes are for collecting data. For example, here they're collecting blood pressure. Do you collect, do you bring the person in, you have them sit for 15 minutes, and then you collect your blood pressure. This allows you to standardize the way you're collecting data and therefore make your data potentially much higher quality for decision making. Another source of data are patient-reported data. So if patients decide that they're not going to actually enter their data on the date that they're supposed to, but they go in at a later date and they enter their data, all at the same time, it's not going to be as high quality. Or if patients decide, I don't want to answer that question today, that's a very valid right that the patient has, and they can certainly do that, but it does affect the quality of our data. Next one. The future is the current status and the future is getting data from electronic medical record extraction. And as you see, as patients are actually meeting with their physicians, really it's important that they also are very cognizant of providing their physician with high quality data. Because a lot of times for these clinical research studies, we go into the medical record and we extract it. So if the data are missing, if the data are not of high quality in the electronic medical record, they're not going to be high quality in our clinical research database. And next one. And with COVID, we now have the new what we think is going to be norm, and that's going to be telehealth encounters. And how are you going to collect data with telehealth encounters? Let's say you've got an obesity study going on, or it's important to collect height and weight. How are you going to be able to accurately do that at telehealth encounters? So I wanted to point out that this is just some of the sources for clinical research studies, but each one of them highlight that it's really important that for each of these sources, we try to get data as valid and as standardized as possible. And you can hit again, I think there's another. So standardization of procedures and processes reduce variability and bias in data, leading to valid decision-making. Next slide. So what is decision-making in clinical research studies? Somebody has a research hypothesis that they want to test. This is just one example. Drug X improves function in patients. So what do? You figure out what's the best outcome measure and what's the best study design for testing that research hypothesis. And this second bullet is where clinical trial readiness really comes into play. And that's what Tiina was talking about. Trying to really understand before you design a confirmatory trial about what are the best measures that are really going to test this hypothesis. What kind of a design should we use? Who should we include in this design? How often should we test these patients? How often should we ask patients to do patient-reported outcomes before they get too fatigued and our data quality goes down? And then what we do is we collect data and make decision. Now this slide makes it seems like it's a very easy process, but it's not an easy process. It's very, very much goes into this, but this gives you the gist of it. Next slide. So how do you make decisions? Decision making relies on statistical testing. And statistical testing relies on looking at what's called a signal to a noise ratio. Next slide. Next slide. And what I wanted to point out with this slide is in the left, I want to show you that the lines in the middle are the signals. So if I'm testing an outcome in two different groups, the green line are the difference in the means between those two groups. That's going to be your signal. So how different on this outcome are these two groups? But I want to point out the difference between these two graphics that I have. The signal is about the same between two groups. In other words, the difference between group A and group B is about the same. But notice in group A, I've got high variability. These points represent the outcome in each individual patient. And there's a very widespread, a very high variability, which makes it very hard to determine, is there really a difference in these groups? What causes this variability? Lack of standardization in collecting data. So my example with the blood pressure, if everybody isn't consistent in how they collect that blood pressure, that can really increase variability. And lots of other sources of variability can really make this spread in the data very, very wide and really hinder our ability to make good decisions. Whereas on the right, you'll see that my variability and my spread are a lot less. And that's because during the study design stage, when I think about how am I really going to conduct this study, I've tried to reduce and eliminate any sources of variability that I possibly can. Next slide. And this is just an example of what statistical testing is all about. So again, you are looking at a signal to noise ratio. And if you, can you hit the slide a couple times? The top, so the difference in the means, like I showed you in that previous slide, that is the signal. And the bottom is the noise. And there's two things I really got to point out on this slide. One is in the denominator, you have the variance. And the variance is what I showed you in the previous slide. How much scatter is there in each group? But what's really important, in addition to the variance, is the bottom is the sample size. How many do we have to recruit in each group? And in rare diseases, this is a very important concept because we just don't have a lot of patients to recruit. So I need to make this denominator, this noise, as small as possible in order to allow me to be able to make a valid decision and have a positive study. And so the only way to do that is reduce my variance or increase my sample size. Next slide. So as I've said, we want our signal to be as big as possible, and we want to really reduce our noise. And the way you get your signal as large as possible deals with clinical trial readiness. What's the best outcome? What's the patient population? What are the best procedures? And again, reduce our noise, and I've talked about that. Next slide.
Eileen, we just have a couple of minutes left. I'm sorry.
Yep, just got a couple slides left.
Cool, thanks.
So again, reducing the variability, make it smaller through design your study, increase your sample size. Next slide. And I think, okay, didn't increase my, okay, so you've seen this slide. Some of you have seen this slide before, and this is usually the slide I use to end my talks. So again, we're trying to get new, better treatments and faster diagnosis because we want to improve the health and wellness of patients with rare diseases and their families. Go ahead and build the slide. As Tiina said, we have lots of people working in their own sandbox. And this sand, go ahead and build this whole slide. Hit it 2 or 3 or 4 times. There you go. And this, if this sand is not consistent so that people can share the stand without having to do a lot of things to it, it's really going to hinder our ability to share data, to learn from each other's data, and to make good decisions by sharing data. In other words, if some of these consortia and some of these teams are using very coarse sand and others are using very fine sand, I may have to do something to the coarse sand in order to be able to combine it with the fine sand. When this has to do with data standardization, and I'm just gonna kind of give you one little example and then I'm gonna end my talk here. Just something very simple, like if I collect gender in one study and one study, one database says, I'm gonna collect gender as male, capital M-A-L-E, and female as capital F-E-M-L-E, and another study says I collected as M and F. I've got to manipulate that data in order to be able to combine that data so people can share data, they can build off of other people's database, they can share tools, and they can build a better knowledge structure. So it's very important to have data standards. That helps with data sharing, and that has to do with how you're collecting your data, but also standardization of processes, and that helps you to reduce your variability. And I think those are all my slides.
Excellent. Thank you, Eileen, very much.
Sure.
So Josh, you were going to show us how you increased your signal, I think, right?
So good afternoon, everybody. I guess good morning still, my apologies. So I'm trying to give a little bit of the perspective from the patient about all of this. You know, what does it really mean to the folks we represent? NephCure, just to really quick go back, is a patient advocacy group, but we're a little research focused. We were founded about 20 years ago with the goal of finding a cure, just like most of us, right? And supporting patients along that journey. And so you'll see on the next slide, so how did we had a group of about 4 people when we started, and how did they go about all of this? And today we're facing literally 24 clinical trials in progress. And so how did that all start? I mean, you know, we're all at different phases in this group. And they were really pretty wise folks. At the very beginning, they went to the NIH and said, okay, help us. How do we, you know, get to close? And so there was a very deliberate strategy. You know, step 1 was, how does this disease tick? You know, what was the data? I'm sure, like most of you, you know, we don't know what causes these diseases, but at least when it happens, what's the pathophysiology, the fancy term, right? How does it tick? What's the pathway by which these diseases happen? Sadly, with nephrotic syndrome, we have 48 different genes that are triggered. And so it's not like cystic fibrosis where you turn off a couple and you can get a cure. It's pretty complicated. So, the good news is that $40 million worth of investment really went into figuring out this disease. How does it work? And the idea is once you do that, then you can go to pharma and say, hey, we have a pathway involvement in fibrosis, inflammation, and those type of things. And they go, yeah, I think I've got a drug on my shelf that may or may not work for that. Let's take a look. So that was step one. Step two is, okay, believe it or not, we live in a society that is capitalist. We have to recognize the fact that while all the studies that we're doing, at a grassroots level are wonderful, at the end of the day, it really comes down to the pharma industry that is responsible for bringing these to market. And I have to shame my bias. I spent 34 years in the pharma industry, so take it with a grain of salt. But I'll share a dirty little secret. They are not philanthropies. They are for-profit entities. And at the end of the day, follow the money trail. And that means, how do I get to have pharma make a profit by studying my disease? Because without that, honestly, it's a tough road. And so what I have to recognize is two things. One, we needed to understand the patient journey. And so the NIH and NephCure co-sponsored studies, and Tiina did a job this morning describing Neptune, which is ours. We would not have made it today to all these trials without that. That observational trial over time really paved the way to understanding the disease. And at that point, you kind of pivot. I wouldn't say you dismiss NIH, but you work with them. But now you open up to the FDA and you got to say, hey, what's the strategy to attract the pharma? How can we help them? And I can't say enough good about the FDA and working with them over the years. Aliza Thompson and Dr. Stockbridge as well over at the cardiorenal division are wonderful people. They really worked hard and developed a biomarker strategy for us that replaced the old way of having to do these big, expensive, and I'll be honest with you, pharmaceutical cost prohibitive approaches. Then we got into now a biomarker strategy, and that led to what you see on the right, which is now we're finally in step 3, which is the clinical trial phase. But here's the key. Once we got into the clinical trials, probably what you face too is that there's a real lack of infrastructure in rare disease for conducting clinical disease. By very nature, rare disease doesn't lend itself well to a lot of clinical expertise. And you're often going to find that the people who are responsible for these trials and the specialties just aren't set up. In our case, nephrology, another dirty secret, out of the 15 major specialties that exist in medicine, nephrology was ranked last. So we had to come in and we started this program called Gateway. And we worked with the NIH, the FDA. We brought all the cross-functional stakeholders together to, and again, all owning this thing called Gateway. And it really was designed to reduce the barriers to recruitment. The goal was no trial should fail due to recruitment problems. If you fail due to, hey, God's going to have half of these drugs not make it and the other half will. But at least don't let recruitment be a barrier. And so we'll talk about that in a little bit. But that was the very deliberate step. It's, in hindsight, obvious, and we're all at various steps of this journey, but that's how we approached it anyway. So let me just talk a little bit about Gateway. As I mentioned, it's a cross-functional, cross-stakeholder global initiative. We brought together folks from around the world, 200 like-minded folks to talk about this. The key is that the focus really needs to be short-term. You're going to find often in academia, hey, we'll put something in place and it may take years to get the result. We don't have that time. And another secret in the pharma industry is most of these companies are venture capital backed, right? And that means time is money. They raise enough money to get to the next milestone. And so if they're late on recruitment, they're going to pull. They're going to pull the trial. And so you've got to focus on how can we change this in the short term. And there may be a lot of great things, But the filter's got to be, if I can't impact recruitment in the next 6 months, it's off the table, okay? So one of the things we put in place is a patient connector tool. And if you go on kidneyhealthgateway.com, and I'll share this with you guys later, but it's our website that basically a patient could go in, fills out a basic criteria, and then the algorithm will spit out, hey, you qualify for the following trials. And the pharma companies help us maintain this with their entry criteria. And then you can click another button. You can connect directly to the trial, or you can get connected to the company or the doctor in your geography based on your zip code. And so those kind of tools is what we put in place, and happy to share, privately or and one-off. But the goal was, again, address the barriers. So I won't get into it, but the key to this whole thing is that you've got to really touch base with the sponsors, the pharma companies. We found that probably half of the protocols that these folks put forward, guess what? The patients don't exist. or the patient burden for the trial is so high. You want me to give how many blood draws a month? You want to give how many urine collections? That's just not going to happen. And so once we sat down with them, you see recruitment, and you guys know your patients, right? You know what they'll stand for, you know what they won't. And, remember, pharma companies try to get the best, the moderate patient without all these extraneous, comorbid situations. And oftentimes they just don't exist. And when you allow those protocols to be in place like that, you're not going to recruit, right? And so they have to realize they have to give a little bit. So that's Gateway. The other thing that was really helpful, this is a program sponsored by the FDA, and if you haven't had one yet, they're wonderful. They're called externally led patient-focused drug development. The FDA is basically saying, hey, help us, not-for-profits, patient advocacy groups, help us find out from the patient view what these trials ought to look like. And I won't go through this list, but please take a look. They're probably not unique to us. But the key takeaways, I think, here is that education is key for the patient and the nephrologist. So let's go to the next slide on that one. So number one, patients, let's face it, there's a lot of myths out there, right? Number one, I'm going to be a guinea pig. You're going to never talk with me. I'm just going to be used, etc. What patients don't realize is that most of it, and dare I say, of all the people on here, right? Think about it. Your disease is not going to spontaneously remit, right? Well, and it doesn't go away on its own. Diet and exercise most of the time won't do it. So what does this mean? It means that most of these trials are not going to be placebo controlled. They're going to have maybe a run-in period where you get a baseline, and then there's going to be an active drug comparator. Or even if it is placebo controlled, there'll be crossover design. And why this is the aha for the patients, they need to know that most of these trials are going to be open label. They're going to get drugged. You don't have to go back on some of these toxic agents that you've been taking with a lot of adverse events. And so that's number one. Number two, a real key is that, guess what? If you respond to the drug and there's no drug on the market for your disease, there's this thing called open label extension, where the principal investigator calls up the FDA and says, hey, I'd like to keep this patient on drug. So no joke, we literally have patients who are on drug for 5 years already and doing great. And the company loves it. They get data on safety. The FDA loves it. The patient, everybody wins in this situation. And that's more the rule than the exception. So open label extension. So these are things the patient needs to appreciate and understand. The other thing is, we did a survey, several hundred patients, and not surprising, and you probably all know this, the number one reason why patients participate in trials is actually right from the heart. I want to help others. I want to give back. So we need to appreciate and remember that if we just explain what's on the left of this slide to them, it marries with what they want to do on the right, okay? Other reasons you can see on the other piece is that I don't want to go back on meds that we're often borrowing. Let's be in nephrotic syndrome, we're borrowing cancer drugs for God's sakes. And the only thing I will say is that last bullet that you see in the lower right to be aware of. Oftentimes you're going to get a lot of patients who have tried everything And they're the folks most interested in trials because they're desperate, they're at the end. Sadly, a lot of these folks, their disease has progressed so far that they're going to be excluded from trials. So oftentimes we'll put them into the non-drug trials and the observational trials like Neptune and others, where they can still give back, still get active care, et cetera. Okay. So, okay, let's talk about the patient now. What are the benefits to the patient other than what we just talked about? They're going to get disease, excuse me, they're going to get access and be able to give back. So here's an aha moment for patients often is that, here's the deal, you are going to get access to the best care in the world. And that's not a dramatic statement, it's a fact. Because think about it, the people who conduct rare disease trials, unlike a hypertension trial, which anybody can participate in, they need thousands and thousands. Rare diseases are typically conducted by the best there are. These are the top specialists at the top institutions in the world, right? And so you're likely to see the best. You're going to get a full workup. And by the way, bullet number 2, it's all free. So you're going to get best access, best care, constant, really hand-holding through this. And it really is something that patients ought to be encouraged to participate in. That third bullet is often not talked about, but another little secret from an industry perspective, the average company pays over $100,000 per patient to participate in these trials because they're hard to find. They have to set up so many sites just to get one patient. And so they typically do provide for transportation, including getting on a plane and flying from Minneapolis to LA or vice versa to participate. So that is usually part of it, but patients need to ask because if they're already spending $100,000 on a trial, what's another few thousand for airfare, right? Okay, so those are from the clinical perspective, I think some pearls that we learned. The final slide.
Josh, not to interrupt, we have like just a couple minutes left before Kristen, okay?
You got it, I'm sorry, I got last slide. Health equity, real quick. It is a contemporary topic that we're all discussing and dealing with in the landscape of America right now. And let's face it, we have a history in this country with Tuskegee, and we have to acknowledge that is not favorable. But right now, the clinical disease, the rare disease is flipped upside down. Right now, it's typically privileged Americans who are having access to these very high institution university settings and getting into these trials. The reality is this is what we call the great equalizer. Regardless of your healthcare plan, you don't even have to have a healthcare plan to participate in these trials. So we've got to make extra effort to get into seeing the minority populations and make sure that they're getting the same access as more privileged folks. So there we go. I think that's my final slide. And so what I would say is if you do this, it's been helpful to us to reach out to organizations who are more, have a lot more, let's put it this way, experience in dealing with minority populations and trying to do it ourselves. They do a wonderful job. So sorry to go long, guys, but hope that was helpful. And again, please feel free to email me if you want any additional information about Gateway or anything else we have. So thank you.
Thanks, Josh. And I know people have questions too, so that was great. Kristen, bring us home here.
Hello, everyone. And Josh, thank you. I swear I learn something from you every time you speak, and I really do appreciate your experience. And I'm kind of in love with your Gateway project. I just dream of there being that many clinical trials in the Porphyrias, perhaps not all at once, but I just think it's so wonderful. And thank goodness for data experts like Eileen, like Josh. I am rotten at statistics, so I so appreciate someone that can understand everything that she presented to us. So anyway, my name is Kirsten Wheeden. I serve as Executive Director of the American Porphyria Foundation. And now you're going to hear from the very other end of the spectrum, the ultra rare. We have within Porphyria, fewer than 12, we have about 12,500 members of our organization, including patients, caregivers, and families, and that's across 8 disease states. So we are far, far smaller than most. I'm going to go through some slides here and share a little bit about the APF, a little bit about Porphyria, and our involvement in clinical trials so far. And then I'm going to share one kind of a case study of a clinical trial that we currently have in process. And I know that so many of you have experiences, and I hope at the end here we have time for some solid Q&A and to learn from others out there who have engaged in clinical trials. But at the AP, we are hands-on with most, if not all, of the patients that are participants in our studies and our clinical trials. So our foundation was founded in 1983, and this is significant because it coincided with the launch of the Orphan Drug Act. And panhematin, which is treatment for the acute porphyrias, was actually the very first FDA-approved drug. And all these years later, it is still a life-altering treatment for patients. At the APF, we are dedicated to the health and well-being of all individuals who are affected or impacted by porphyria. As I mentioned, we have about 12,500 members, including, again, patients, caregivers, and families. The chart at the bottom left of my slide here, I'm a little bit in love with, because each one of those dots represents a patient with porphyria, color-coded to the different types of porphyria. It just always amazes me where our patients are located. And it also just motivates me because I want to impact every single one of them are out there. Now that patient outline, it aligns nicely with the map to the right, which is the location of our Porphyria centers and our satellite centers of the Porphyrias Consortium. That's where our experts are. We have 6 Porphyria centers and 8 satellite centers of experts and specialists. They are brilliant. They are leaders within this fight. They're incredibly accommodating to the APF and to our patient community. Now, on the bottom right side there, I've listed our 5 pillars that we live by and work by every single day at our foundation, and you'll see that research is at the top. Since the beginning of the APF, our motto has been, it actually first appeared in our 1984 newsletter, our motto, research is the key to your cure. And we are laser focused on trying to move research projects forward, both observational studies and the clinical trial. We really believe that we are nothing as a united community if we cannot come together to really support and move forward the critical research. Let's see if I got there we go. All right, this looks rather complicated, but I'm going to give the shortest possible explanation of porphyria to lay a little groundwork for our participation in clinical trials. So porphyria is a group of 8 ultra-rare diseases. They're all based in the development of heme. So to simplify, there are 8 steps in the development of fully formed heme, and every time there is a misstep, it equals a different type of porphyria. So the porphyrias are typically divided into 2 main categories, acute and cutaneous. The 4 acute porphyrias are neurovisceral diseases. Picture nerve endings that are bathed in neurotoxins, and patients then present with symptoms such as horrific abdominal pain, nausea, vomiting, tachycardia, brain fog, or mental confusion. It is known as one of the most painful diseases in humankind. Then we have the 4 cutaneous porphyrias, which present with extreme phototoxicity. And these are the group of diseases I'm going to focus on mainly for the next few minutes. In these diseases, the sun and oftentimes artificial lighting cause extremely painful reactions upon exposure. Now, on my slide, the 8 porphyrias are listed from top to the bottom, and it's based on the defect in the heme development. Mother Nature did not put them in a lovely order for us, as you can see. There are 4 cutaneous, 4 acute, and they are all mixed up in the pathway of heme. And even 2 of them have dual symptoms to confuse it even more. But the real intention of this slide is to show the treatments that have made the journey through in the porphyria community. As I already mentioned, we were blessed to have the first FDA-approved treatment, which is panhematin, a biologic. That was in 1983, and it's only for the 4 acute porphyrias, which are indicated by the brown arrows. Now, since 1983, it's a lot of years of life-altering treatment, but I will say that we have fought and we have clawed for access for patients for this treatment. It has certainly not been an easy road. The next treatment to the right is Givlaari, which was approved in November of 2019. It was actually at our CPAG meeting. We were in the middle of the meeting when I got the information that it had been approved. This is an RNAi treatment through a monthly injection, which reduces ALAS1 in the liver and reduces the number of attacks patients will have. There were 94 patients in that clinical trial, and the APF recruited each and every one in the US portion. And there are now nearly a year later, we have about 100 patients in the US that are being treated with Givlaari, which is enormous and again, life-altering. The next one to the right is Scenesse. Scenesse is a bi-monthly implant to prevent phototoxic reactions in patients with EPP and XLP, 2 of our cutaneous porphyrias. The APF was the central node for the Scenesse phase 2 and phase 3 studies back in 2011 and 2013 that was finally, and I emphasize finally, approved last year on October 8th, 2019. That was an enormous day of celebration after a decade or so of advocacy. So for that clinical trial, imagine those patients that had that taste of freedom, were able to be exposed to the sun and to live a normal life, only to have it taken away. It was not they did not have the benefit of an open label extension or compassionate use, and they waited years for approval. Now we have many, many patients who are being treated in the US. And then lastly, on the right-hand side, we have MT7117. That is currently in clinical trials for that same group of patients, I might add, for EPP and XLP. This is a daily oral treatment, and we have successfully navigated through phase 2 clinical trials. I want to mention that I just find it in our experience that of these 4 treatments, we have an infusion, an injection, an implant, and pills, 4 mechanisms of delivery. And of these treatments, some are used to, or a couple are used to prevent attacks, and others are used to treat attacks. So it's very different, a very different clinical trial experience for each of them. I'm going to focus a few minutes on engagement and collaboration. Josh did go over much of this. It'll be, but I think it bears repeating because I think to have a larger clinical trials and smaller clinical trials, to have the engagement and collaboration at the core and key to each of them is important. So there are many players in the field, in this field, to get a treatment from the lab all the way through FDA approval. The patient advocacy group, I believe, or we believe, plays the role of the glue that keeps this together and moving forward. It can't move anywhere unless there's someone speaking on behalf of the collective patients and engaging and educating all of these stakeholders listed here on the true nature of this disease state, hopefully with a valid natural history study, and sharing what is a meaningful outcome to these patients and how we motivate patients to participate in clinical trials. If our job is done well, really well, we have gathered that patient information and proved to the sponsor or to industry a readiness to go to clinical trials. And as Josh said, they have spent millions by time to get to this point, and they need to know that we have a community that is ready to participate. So patients, we are always saying, left and right, everywhere we go. Patients come first, because as you all know, so often patients come last. So we have worked hard to help all to decrease the burden on patients to participate in clinical trials. More home visits, an educated concierge service, study sites that are close to home, which means more study sites, access to expert care. We do a lot to both decrease that burden and educate the patients that we are decreasing that burden. The FDA, we have also been blessed to have many meetings with the FDA. We have held 2 patient-focused drug development meetings, both internal and external. They were terrific events. We had a scientific workshop on erythropoietic protoporphyria in the Great Hall on the White Oak campus. I'm sure most, if not all of you, have been in that room. It is a very large room. As I mentioned, this disease state, people cannot be in artificial lighting. So picture that enormous room in the dark. We had our meeting in the dark. They even had to shut off the monitors on the screens because people had traveled to get there to get their voices heard, and they were singed from being in the sun just to get to that meeting. We also held a rare disease listening session. This was held last August, which was a tremendous event, a much smaller event, and we were able to highlight the complexity of each type of porphyria. Later in that year, we had 2 drug approvals on our hands, so we really feel through all those that our voices were heard at the FDA. Now, we work really closely with industry and their contract research organizations specific to each clinical trial. And we play a formal role with them for clinical trials, and we have facilitated their engagement with the patient community through patient meetings, roundtables, advisory boards, but we are at the center of that process. And then last but not least, we collaborate very closely with our key opinion leaders who advise the industry partners and also participate as clinical trialists. This trust has been built over years of working together. And I would be remiss if I didn't mention that I do believe that research coordinators make the world go round. Their ability to communicate and to organize the patient communities, we cannot herald them enough in the critical role they play in the success of clinical trials. So after you've developed the critical research-ready cohort, we go through a process of educating our patient community, and then a specific process to refer patients to clinical trials. We take a lot of time and effort to educate our patient community on the drug approval process. The process requires long-term patient involvement, a lot of patience, and an incredible effort from a patient community, and I would say specifically in rare disease. Participation in research is the only way. I mean, it's the only way we're going to move forward with access to treatment, let alone a cure. In fact, we call our patients who participate in trial medical heroes, because truly believe that's what they are. So education efforts begin a long time before a clinical trial takes place. We share information constantly and in various ways about the phases of a clinical trial, the FDA approval process, and then even more detailed education on things like what is a placebo? What does placebo-controlled mean? What is double-blind? What is a double-blind, an extension? What is randomization? All the words that we hear every day and are meaningful to us, but maybe a little bit scary to a patient unless they're fully explained. Then it is up to us to know a trial inside and out, to manage all the questions that are going to come our way. And within that process, we are very, very careful never to offer medical advice. We communicate our IRB approved materials across all our platforms. And then because we are a tight-knit community and our patients really now come to expect this of us, We talk to each potential participant one-on-one, explaining the details of a clinical trial and answering questions. And we always, of course, we can refer them to clinicaltrials.gov where all this information is laid out. Once they express interest in participating in a clinical trial, we then refer them to a study site and ensure that a connection is made with that study site and the research coordinator for detailed follow-up. We then become the problem solvers because, as you know, there are always going to be issues, challenges, and problems. It will be the study site didn't call me, the car didn't show up, I got the placebo, I'm not sure I want to participate, I want to change study sites, maybe the patient didn't input their data, or a study site visit was changed and now they don't have enough meds. We always encourage them to reach out to the study site for resolution, but they seem to come back to us, which is great because then we can help resolve the issues and work toward a successful end game. But if you name it, we'll hear it. And then lastly, the advocacy. That's where the work really begins. If a trial has proven effective, engaging with the FDA, and even after approval, your work moves to access to treatment and insurance approvals, and that entire process rare disease still needs to be understood by the stakeholders even after you have a drug approval.
Hey, Kristen, I hate to interrupt, but we're at 12:15, so
Oh my goodness. Okay.
Questions?
Yep. Okay. So what I'm going to do then is what I was going to go through a particular case study within CNES, within EPP. But I think I'll really just skip through that because I think that even though this was, really important to our success in getting CNES approved, and then now we're in the middle of a trial for the same with MT7117. I'm going to skip through explaining this disease, and anybody who would like input or to learn from our experience specific to EPP, disease that was very complex with issues with a meaningful endpoint. I'm happy to discuss that offline or in questioning. So, okay, so actually, since we have about 15 more minutes.
Yep, and I know we wanted people to, especially for you and Josh, to ask questions.
Yeah, okay, so I'm just going to mention about this trial that we, for MT7117, that we are currently in the middle of, that a couple important numbers on here. 102 people that we needed to recruit for phase 2. We actually needed, by the time we got to get to that 102, we talked to about 240 potential participants who then referred to study sites, and based on exclusion criteria, and other challenges, to get to the point is to get to that 102, we had to talk to many, many more. We were successful. That trial, phase 2 trial, was published and presented at a major medical conference and paved the way to move to phase 3, which we're in the very middle of right now. So I will end with this. We know that the number one reason, or we understand anyway, that the number one reason the clinical trials fail is lack of participation. To us, that is not an option. Patients are suffering out there with these ultra rare diseases that impact every moment of their life. Researchers have dedicated entire careers to helping these patients and understanding these diseases. Industry spends millions of dollars to get here. So by the time it's in the hands, our hands in the patient community, we cannot let it fail. And research is the key to do that. So thank you. Everybody, I appreciate your time. I hope some of that was a little bit useful.